Hello everyone. We can now go over B cell maturation and how we can track normal B cell maturation by flow cytometry. So B cell maturation starts in the bone marrow, and there are different phases of maturation, and it completes in the lymph node. So the early B cells, or the pro B cells, or the immature cells, or the precursor cells. They start with being CD34 and TDT positive, which are the immaturity markers, and these immature markers also express CD10. And one thing that B cells consistently express throughout their life is CD19. So CD19 is expressed even in the pro B cell stage. And that's the only B cell marker that is expressed at that stage. And then they start acquiring CD79A and PAX5. And at that stage, they do not have their light chains or heavy chains or light chains rearranged, and they do not also express CD38, and then they slowly start acquiring CD38 as well. And now when they go through the stages of hematogones, like early hematogones, intermediate hematogones, late hematogones, they slowly start acquiring CD20, and late hematogones start expressing bright CD20, and then they express CD20 from that point till that point throughout their stage unless they become plasma cells. And now, when they come out of the bone marrow, they come out as an immature B cell, and they have just rearranged their heavy chain as IgM. So they are naive B cells, and they have IgM and IgD on their surface. So they go and sit in the mantle zone. That's why in your lymph nodes, when you do IgD staining, you can highlight the mantle zone very nicely. So initially they go to the mantle zone, and then they go into the germinal center, where they are exposed to the antigens, and they have their somatic hypermutation and rearrangement of class switching, and then they start expressing other heavy chains, and they then they evolve into becoming memory B cells and other stuff. But during that phase, they also have certain difference in their antigen expression. The follicular center B cells will start expressing CD10 and BCL6 and other follicular center markers like HGAL and LMO2, which are mostly by immunohistochemistry. Again, CD38 expression is positive in the follicle in the germinal center or germinal center B cells. And then from there, when they decide to become plasma cells, they start losing CD20, they lose PAX5. They still have their CD19 and CD79A, and then they again get brighter for CD38, and they are now positive for MUM1, BLIMP1, and CD138. So plasma cells do not express PAX5 and CD79A, but they and also PAX5 and CD20, but they express CD79A, 19, and nice MUM1. So that's why we use MUM1 or CD79A and 138 for staining our plasma cells in tissues. And some of these we don't use MUM1, but bright 38 and 138 can be used to isolate plasma cells in flow cytometry. Now we can actually track the maturation of especially the hematogones in flow cytometry. Hematogones are the early precursors are increased in pediatric population, and they are also increased in post-transplant patients or in a regenerative marrow post-chemotherapy. So you will see them in the hematogones gate that is just below the blast gate. So this is our lymphocytes, which express the brightest CD45 and low side scatter. We have the monocytes here, the granulocytes here, and the blasts sit just below the granulocytes here. And the hematogones gate is located even below the blast gate, and it kind of is you see the different stages of hematogones, the early and intermediate hematogones sitting here, the late hematogones sitting here. The late hematogones kind of go and merge with the lymphocytes. Sometimes you will see a continuous population going into the lymphocytes, and these are the mature lymphocytes. The early hematogones express CD34, but as they mature, they lose CD34, and they express bright CD38, and they start losing CD38 also. And same way, they also gain HLA-DR and start losing HLA-DR during during their maturation. CD19 is expressed throughout, and CD20 is acquired as they mature, especially they are brightest in the late hematogones, even brighter than the mature B cells, and mature B cells do express CD20. And one interesting graph that usually most people use to identify hematogones is CD10 versus CD20, because in this plot you can see the early hematogones expressing bright 10, then they kind of lose the 10, get a little dimmer, and then they start acquiring CD20. And as they acquire CD20, especially the late hematogones express very bright CD20. As they acquire CD20 and become mature lymphocytes, they lose CD10. So you can see that CD10 is being slowly lost in the hematogones as they mature into B cells. And this is where also the light chain expression light chain expression can be appreciated. These mature B cells will start expressing kappa and lambda, and that's what you see here in in flow cytometry also. So immature cells do not have surface light chain expression. As they mature, you can see them expressing light chains. So all these immature cells are negative for surface light chains. Only the pink population or the mature B cells express surface light chains. So it is very important to know the immunophenotypic pattern of hematogones and various stages of their maturation, because especially in bone marrows that come for follow-up after treatment with BALL, you will have sometimes very will face a lot of challenges to differentiate between residual or minimal residual disease blast population and to separate them from the normal hematogones. So it is very important to know their pattern of maturation with different markers so that you can kind of differentiate the two populations. One clue is that hematogones are a dynamic population, which means they are maturing. So they are not going to be clustered in just sitting in one place. They are going to be a dynamic population, so you can actually see them in different phases of their maturation, unlike the blasts, which are tightly clustered and they are going to be there. You will not be able to track them mature into something else. So that helps. So that's about B cell maturation. Thank you.